Welcome to Masai University. My name is Eric Ogello, the Department of Fisheries and Natural Resources. I'm glad to welcome you. In Masai University, in our Department of Fisheries and Natural Resources, we are doing teaching both undergraduate and postgraduate students. We are also doing research, and we are also doing community service. So in terms of research, we are doing specialized research in our laboratories, in our fish farm, and we are also doing community research, that is in adaptive research. Some of the projects that we are currently doing, we are doing bioflow production, and we are also doing fish health management and coastal aquaculture. It refers to Artemian production. I'm going to show you in our laboratory where we are doing specialized research in labs and in also our field fish farm, where we are culturing fish, both tilapia and catfish and also labeo. Some of the fishes we are culturing are actually meant for restocking the lake and also restocking the dams so that we can have fish sufficiency and fish abundability for everyone in this region. So follow me into the laboratory. This is our laboratory where we are doing research on fish in Lake Victoria. We are also doing research on aquaculture. Masai University is located just at the foot of Lake Victoria, and we are doing a lot of research to ascertain what kind of fish we have in the lake, what type of fish we have there, how they are growing, and how people use those fish for their daily needs. So quickly, let me start by saying that initially Lake Victoria had a lot of native fishes, and most of those fishes disappeared because of the introduction of Nile perch. And just what I'm carrying on my hand, this is a lungfish, one of the fishes that disappeared from the lake because of predation from Nile perch. What we are doing in our lab is that we are trying to map the genetics of the old fishes that were there in the lake so that we have that gene pool with us. At the same time, we are also trying to, study fish. For example, this is Nile tilapia, or Oryctolagus niloticus, which is very much loved by many people in aquaculture. So what we are doing here, we have actually preserved the old Nile tilapia from Lake Victoria, and this is a gene pool of this fish, so we can track the genetic changes of fishes that has occurred over time. So we are actually doing some experiment to determine if the current Nile tilapia has deviated significantly from their ancestors. We are also actively engaged in other native fishes like bacchus. You can see this is one of also the native fishes, bacchus dogmark, or locally known as seu. These fishes have a lot of native attachments because they are local delicacies. But right now we cannot find them. So whenever we see some of them coming back, we try to see if their genetic makeup is different from the original one. Microscopic work, yes, we are doing water analysis, plankton analysis, both phytoplankton and zooplankton, and also macrobenthos. And sometimes we use this lab to analyze, for example, samples from the bottom of the lake, surface of the lake, and even the planktonic region of the lake. So like here, I have samples which I fix, and once I fix it, these are samples from our fish ponds. So I do check the concentration or the density of the phytoplankton life in terms of rotifers, copepods, and cladocerans. You remember, these are live food materials that are very, very important for survival of larval fish. And we have been able to promote live feed as very important first diet or first nutrition for larval fishes because we see significant reduction in terms of mortality. And most of our farmers say that they experience a lot of mortalities because of the kind of diet they are using to feed their fish. We are also manufacturing feeds. Just on my hand, these are pellets, 2mm pellets. So our students can formulate diet using local materials and produce feeds which are used for feeding fish in the pond. We are not buying feeds, we are producing our own feeds. Welcome to our small hatchery. In this hatchery, we are producing fingerlings for aquaculture. We are also very active in aquaculture, and the main problem we are facing in Kenya in aquaculture is seed production and feed production. Here, we are nurturing live fingerlings for tilapia, and we are doing sex reversal because farmers are now using monosex, that's all male tilapia. So we are using the 17-alpha-methyl testosterone hormone to sex reverse fish, and once they attain reasonable size, we actually roll them out to the farmers who stock them and grow them to maturity. So you can see here, we have, this is pure Oreochromis niloticus, and that can be evidenced by the clear bands that we are seeing, and the way the tails have been also been marked by the spots. So here, we feed them here. Now these ones have already been sex reversed, and they are all males. And you can see they are also all in uniform size. So once we have attained the sex reversal, we actually dispose them to farmers. In this laboratory, we also use it as a fish bank, where we keep fish from the lake and also from the ocean. So essentially, this is our gene bank. So here we have a gene bank of fish from Lake Victoria, and these are different species. This is a very interesting one because Lates niloticus is one species that is being accused for eating all the other freshwater fishes in Lake Victoria. And now we are in a situation where we have very less fish in Lake Victoria. Now what we are doing here, we are storing this gene bank and we are using it to study the scientific generations of fish. For example, the fish that we have in the lake right now, we analyze the genetic material and we compare with the ancestors to find if there are impacts that have occurred in the genetic makeup. And that's very useful for us also to determine if maybe climate change has affected the genetic material and genetic diversity of the fishes. So we have fishes from Lake Victoria. You can see we have the Lates niloticus, we have the Cichlidae, and we have the Falciformes, also Muchochilidae. These are actually native fishes that no longer exist in the abundance in Lake Victoria. We also have marine fishes. I say, being a gene bank, it's also interesting to store some of the interesting marine fishes, like the millfish, the butterfly fish. We also have the porcupine fishes, and the common name. This is actually pufferfish. This pufferfish is very poisonous and also very nutritious. So basically, we have stored their genes here, and we are using it to study. And you can see we also have our students. So the students are in a session. We are using this fish for their day-to-day experiments and life. So from here we are going to move to the fish farm, where we are now going to see how we are actually doing aquaculture to promote fish diversity and fish production. This is our fish farm, Maseno University Fish Farm. The ponds, you can see we have concrete ponds, and all of them have broodstock. We have tilapia broodstock, and we also have catfish broodstock. We are also having labeo in this farm. So this is where we are breeding fish, and once they have produced fingerlings, we actually transfer either the eggs or the fry into the lab for further incubation. Now this farm is very important even for this community, because this community find the fish seeds here, and they also use this farm for training. So they come and we train them here. Aquaculture is very critical for food security, because you know if every person has a pond, backyard fish farming, aquaculture replaces fish capture fisheries that has now plateaued in terms of production volumes. So that's why aquaculture is interesting in food security, to improve people livelihood of people, by reducing malnutrition and improving economic well-being of small-scale traders and small-scale fish farmers. And that's why it finds its place in the food systems, which is now the buzzword in the food industry, not only in Kenya, but the entire world.